Previously on GBE 403 Term Project.
The cell cycle is a process of DNA duplication and division of cell to produce two daughter cells. That includes many processes for successful self-replication. Understanding with this, scientists identified the four characteristic phases of the cell cycle, such as mitosis, gap1, DNA synthesis, and gap2. The event of DNA replication, chromosome segregation, and mitosis define a fundamental periodicity in eukaryotic cell cycle. Basically, even though the details of the cell cycle vary from organism to organism and at different times in organism's life, certain characteristics are universal.
In mammalian cell cycle studies like this one of the prime objectives is to identify cells in the process of genome replication and consequently incorporation of radioactive DNA precursors such as tritiated thymidine has been one of the main tools for such analysis.
This study elaborates the determining cell cycle and the effects of growth inhibiting factors on proliferation. The reason to choose this study is to acknowledge more information about regulation of cell cycle during proliferation.
The study also gives example of how the method can be used to measure the effects of different growth inhibiting factors.
Hello everyone! In this lovely Sunday evening, we had some technical issues, so we are a little bit late this time. As we all know, this is the last video. In this video, we will talk about how to analyze the data obtained from flow cytometer and all kinds of details. Additionally, we have some representative results for you. In this experiment, efficiency depends on a few key controls and experimental conditions, such as control samples. This control serves three important purposes. First of all, it ensures that culture conditions used for all experimental samples are enough to support proliferation constantly in the absence of treatment. Also, this sample is needed as positive control to be sure that BRDU and CD20 positive cells can be detected when they are present. And the last purpose of using this sample is provide the calibration of flow cytometer to have reasonable results for other samples wanted to be analyzed. This control sample can be used to adjust the sensitivity of photomultiplier tubes for propylium iodide staining such that the 2N and 4N peaks from singlet cells are centered at 200 and 400 on the x-axis, and the sensitivity of photomultiplier tube for FITC detection such that G1 and G2M populations. So, negative and positive signals are centered as shown in the figure. This is quantitation of cell cycle phases by combined propylium iodide and BRDU staining. This panel shows three-dimensional flow cytometry of propylium iodide and BRDU stained cells. Cells with 2N and 4N DNA content are centered over the 200 and 400 marks on the x-axis scale for propylium iodide staining intensity. BRDU staining intensity is measured on a logarithmic scale on the y-axis. The position of gates used to quantitate cells in the G1, S and G2M phases of the cell cycle. And this is the relative proportion of cells in each of the G1, S and G2M gates from previous graph.
These examples demonstrate how cell cycle control can be measured in response to a variety of stimuli or cell manipulations. This approach can therefore be adapted to many applications that require the measurement of cell cycle positions in culture-type experiments. We have three examples of experimental cell cycle analysis. The representative results also make other important points. First of all, they demonstrate that BRDU uptake and labeling can vary between cell types. For this reason, it is important to empirically determine the length of pulse and staining conditions needed to detect cells in S phase. In general, cell types that double in culture in 24 hours or less can be labeled with BRDU in 1 hour. Slower growing cell types may require longer pulses and alterations to antibody staining conditions and durations. The first use retroviral expression of the cyclin-dependent kinase inhibitor P27-KIP1 in mouse embryonic fibroblasts, 24 hours after drug selection for viral infections was complete, cells were pulse labeled with BRDU for 1 hour. In this experiment, ectopic expression of the inhibitor is used to arrest proliferation of cells. Little BRDU positive events are evident in S phase gait in response to P27. Likewise, the percentage of cells in S phase for P27 expressing in cells is quite low and diagrammed in this figure. This type of analysis has been very effective in characterizing of cell cycle control defects in cells derived from various stains of gene target mice.
In the second experiment, untransformed mammary epithelial cells were treated with the growth inhibitory cytokine, transforming growth factor beta-1 for 24 hours. Cells were labeled with BRDU for 4 hours immediately prior to harvesting, as shown in the figure. BRDU labeling in S-phase cells is greatly diminished by TGF-beta-1 signaling. The specificity of the staining is also validated with an IgG negative control as seen in the figure. Quantification of the different phases of the cell cycle confirms that TGF-beta-1 primarily inhibits proliferation in the G1 phase of the cell cycle, leading to an accumulation in this phase. Strong G1 accumulation creates essentially a single G1 peak that could be misinterpreted as G2M. In our last example, PRB deficient SAOS2 cells are transfected with an expression vector and either CMV-RB or CMV-beta-GAR as a control. 3 days following transfection, cells were harvested, stained and fixed. Flow cytometry analysis of these cells is shown in the figure. This demonstrates the cell cycle distribution of negative control transfected cells in comparison with the distribution following 72 hours of PRB expression. Following curve fitting by multi-cycle software, a direct comparison of the proportion of cell cycle phases is shown in this figure. This reveals the accumulation of cells in G1 following PRB expression and relative depletion of cells from the S and G2M phases. Variations were used on this assay to probe G1 arrest mechanisms extensively.
One limitation of these types of flow cytometry approaches is that they are only able to establish relative abundance of cell cycle phases compared to one another. For this reason, it is ambiguous if expression of P27 in this figure truly induces an arrest in G1 and G2M, or if it just slows progression through these phases relative to S phase. These possibilities can be investigated further by treating a parallel sample of cells with a mitotic inhibitor such as nocadazole or G1S inhibitor like afidicodin. Since these drugs create a dominant arrest in M phase or early S phase respectively, slowly proliferating cells will accumulate at the drug-induced arrest point. For example, cells arrested in G1 because of PRB expression will remain in G1 despite nocadazole treatment, while controlled cells will accumulate in M phase. Taken together, these experimental approaches offer a flexible methodology that can be applied to a wide range of mammalian cell cycle research questions. They can readily detect alteration in cell cycle progression and quantify difference when compared with controls.